Welcome to Chapter 15, Part 1b, the vascular system. We left off last time talking about the sort of two sides of blood flow and the role of different parts of the vascular system in ensuring that that blood flow occurs. In order for blood flow to occur, you have to have pressure. So on the left side of this figure over here in the heart, and there are actually two circuits here, there is the right side of the heart and the pulmonary circuit, and the left side of the heart and the systemic circuit. In either case, though, and I'm just going to circle the left side here, even though the right side is responsible for pressure. I'm just going to circle over here on the left. So this side right here, the heart, as well as the expansion and contraction, the elasticity as well as the muscularity of the arterial system contribute to pressure. You can't have flow without a pressure gradient. So this part here and this part here both contribute to the pressure gradient more than they contribute to, say, resistance. Okay? On the right side over here, primarily here in the arterioles and meta-arterioles and what we call the pre-capillary sphincters, which we'll talk about later on in this lecture, they are responsible for modulating resistance in order to control where the flow occurs. The pressure drives the flow, and the resistance over here drives where the flow goes. Now we can have shall we say, deliberate resistance in the form of blood flow control by these arterioles and meta-arterioles and precapillary sphincters, or we can have unintentional control of flow due to other factors that affect resistance, and that can include pathology. If r to the fourth power is inversely proportional to flow, anytime we change arterial diameter, we're going to affect resistance, whether it's quote unquote intentional or not. The last piece of the equation has to do with volume. So I've talked about pressure, and basically to increase pressure, we insert volume into the system. We can also insert volume into the system down here at the bottom in the veins. Like I said last time, 2/3 of our blood is actually in our veins right now. If the large veins contract, they send more volume to the heart, which in turn will squeeze harder and send that blood into the systemic or into the vasculature, which ultimately affects pressure. Okay? When we insert volume into the system, pressure rises. There are some other corollaries to this. This goes in a few different directions. Like, for instance, unintentional vasodilation would cause pressure to drop. Instances like that, whether we lose blood because of a hemorrhage, would cause the blood pressure to drop. Psychogenic shock, toxic shock, all those things that contribute to vasodilation would also cause pressure to drop. So it's a complex system, and you have a few balls in the air. The key is to work through the factors, and we'll go over this a little later on. Work through the factors individually and understand how each factor can influence overall pressure and flow. Just a little bit of anatomy today in this lecture, talking a little bit about the differences between the various sectors and structures in the vascular system. Talk about the different cell types, the different types of tissues that you find in different areas, because this structure plays directly into the functions, in some cases functions that I've already discussed. First of all, notice that each sector, each structure in the vascular system contains endothelial cells, and this even includes the inside of the heart. The inside of the heart also contains a layer of epithelial cells called endothelium, exceptionally important in terms of signaling and in some cases disease. Turns out inflammation and dysfunction of the endothelium is the very, very earliest step in vascular disorders, primarily hardening of the arteries or atherosclerosis. In the artery, we see a lot of elasticity. That's why we have a lot of connective tissue that's relatively elastic, as well as a very robust layer of smooth muscle. So when the systolic wave comes into the artery, it will stretch, the smooth muscle will relax, and then it will rebound by a combination of elastic recoil and smooth muscle contraction. The smooth muscle in the arteries is tonic smooth muscle. It stays contracted and is transiently relaxed. Yes, and there's an outer layer of fibrous tissue as well. Not terribly elastic, but it actually, that's one of the major ones that begins to get stiffer first and tends to prevent elasticity in older individuals. Now we get to the arteriole, and this would be arterioles and meta-arterioles, pre-capillary structures. Very, very small, okay? Orders of magnitude smaller than the arteries, and I'm not worried about these numbers over here, by the way. What we have are those endothelial cells yet again, and you can see them in purple here. Those purple cells are the endothelial cells and smooth muscle. The primary role of the arteriole is to regulate resistance and thus regulate flow. These are the drivers of differential flow to meet metabolic need or physiologic circumstances. So, for instance, if you've had a big meal, what happens is you shunt blood away from the skeletal muscle and towards the digestive tract and the liver. If you are exercising, you will shut down blood vessels going to the digestive tract, maybe a little bit not so much to the liver, but then you will also expand and dilate to the skeletal muscle. If you're sitting in class, various areas of your brain are currently vasodilated. The areas of hopefully listening and observation and memory are all vasodilated, where other types of areas of the brain are probably somewhat normal or even perhaps a little vasoconstricted. Then we get to the capillaries, which we're actually going to talk about capillaries a little bit at length later on. Capillaries only have one cell type, and that's an endothelial cell. They're very, very, very thin. Simple squamous epithelia, very, very thin. This is an exchange epithelium. These things damage very easily and are also repaired and grow very easily, but they only contain endothelial cells. We get to venules, and venules tend to be a little bit stretchy. If you've ever seen varicose veins or that sort of a thing, they probably originated as venules. Or small veins, if you will. Again, endothelial cells and some fibrous tissue. Lastly, the big veins. These are the great veins, if you will. These are the ones that you can see under your skin, or the ones that are the final stages of traveling back to the heart. You get a little bit of smooth muscle, especially in the very, very large veins. You don't see it. If you were to look at the veins on the surface of your hand right now, if you can see them, there's not a lot of muscle in those veins. Those are actually more structurally related to venules. But as you get closer and closer to the heart, the amount of smooth muscle and elastic tissue goes up. Why? Because the veins are a volume reservoir. If arteries are a pressure reservoir, you can write that down right here, a good spot for that. The veins are a volume reservoir. If this large vein contracts, less volume can be stored in those veins and returns to the heart to enter the systemic circulation, causing a rise in blood pressure. Okay. But the key takeaway is that every structure has endothelial cells, and the tissue composition is directly related to specific function. Add just a little more detail about veins. Veins, because of the fact that there's so much resistance once you hit the capillaries, that overall pressure will drop and flow will drop. Veins have developed a couple of differences to facilitate an increase in pressure and thus flow. Veins are a very, very low pressure system and are susceptible to gravitropic or I would say gravitropic type situations where you'll get blood pooling. This is one of the reasons why if you fly on an airplane, they start serving you drinks about half an hour onto the plane. And if you're on a long haul flight, 6 hours or more, 8, 10 hours, they will serve you meals, and travel advisory sites will recommend that you get up and exercise a little bit just to get the blood moving because it won't. The pressure is so low and affected by gravity. In any case, veins generate pressure with two specific mechanisms. One is through muscular contraction. That's why if you get up out of your chair and you start walking around, more blood returns to your heart by virtue of the fact that when these muscles contract, they squeeze these veins and increase pressure, which causes flow. Okay. Secondly, veins have specialized valves that are there to prevent reverse flow. They're basically specialized internal structures. I once saw the medical term one time. It's not important. These valves are there to prevent blood from just settling back down gravimetrically. And unfortunately, these valves are susceptible to failure. So they work great for a while. If they get stiff and they don't close as well, you'll see more and more gravitropic effects. And that's why you see sort of age-related changes in vein structure that are of cosmetic and or in some cases pathologic significance. We talk about things called deep vein thrombosis. Folks, there is an age-related risk, but there's also, by the way, and I'm going to talk about this later, there is a gender-related risk as well, and a genetic-related risk. But we have these veins, the way they're situated in the muscle contraction generates pressure to cause flow, and then these one-way valves when these muscles are relaxed to prevent reverse flow. It's a similar concept that we see in our hearts as well. We have talked a little bit about exchange in the form of diffusion and a little bit about exchange in terms of paracellular and transcellular. And we're going to spend more time on that in the next unit, in Unit 4. Nevertheless, we are in the vascular system, so we do need to talk about the idea of exchange taking place. And the site of exchange, bar none, is in the capillaries. All of the other cell types are not well suited for exchange by virtue of distance. Distance is a critical factor. That diffusion rate is directly proportional to distance. The farther the distance, the slower the diffusion rate. So all the other structures are not well suited for exchange except the capillaries. They don't have vascular smooth muscle. They don't have elastic tissue. They are very, very, very thin, which means that they can allow and are designed to allow exchange, both transcellular as well as paracellular. Now that brings up a good point. In the blood-brain barrier, which is the cellular protection system for the neural tissue, you basically only have transcellular transport. That is to say, any nutrients, any gases, any molecules must pass through those cells that are comprised of the blood-brain barrier into the cerebrospinal fluid. There's only a couple of places where you have basically just areas of the central nervous system that do not have a blood-brain barrier. And the hypothalamus is one of them. Okay. Why? Because it's involved with monitoring the blood chemistry, so it needs to be able to sense these things. In any case, the blood-brain barrier is composed of capillaries. It's just that those capillaries are much, much more tightly interlocked. They have tight junctions and they grow in such a way that there is very little or no paracellular transport. Okay, most capillaries in our body allow significant amounts of paracellular transport. somewhere along the lines of 3 liters or more in excess of what we return back to the blood system per day. Again, I'm getting ahead of myself a little bit, so I'm going to stop there. But in the blood-brain barrier, we have these cells called pericytes or pericytes, as opposed to the parasite like the pathogen. Pericytes, we're not really sure where they come from. We don't really count them in the general lexicon as glial cells. That's why you didn't see pericytes when we talked about the nervous system. They seem to be a combination of vascular cells, but there is some evidence to indicate that they might derive from microglia. So they may have glial lineage, and we just haven't categorized them correctly or something along those lines. The combination of pericytes as well as the astrocytes, they will secrete signals that will enhance the barrier function of the capillaries. So they can differentiate into endothelial cells. They can also differentiate into smooth muscle cells. So again, we don't really know where to put them. And that's one take-home lesson from this class, is our classification systems are not perfect. We often learn new information, which then changes what we think about where things come from or where they go. Where our classification systems are fundamentally model systems, and the model systems are not perfect, and this is a classic example of that. So pericytes contribute to the blood-brain barrier by enhancing the barrier function of the capillaries, decreasing their permeability. They can also differentiate into vascular cells in the central nervous system. Because blood vessels are so critically important to meeting metabolic need in a tissue, blood vessels can and do retain the ability to grow, develop, and change their structure to meet metabolic need up to a certain degree. That process is known as angiogenesis. And we can see a before angiogenesis down here, a tissue that has demand for additional blood supply and nutrients and oxygen can induce that set of blood vessels to grow and expand dramatically. Again, angiogenesis. If you have an angiogram, what you're doing is you are taking a picture or getting an image of the layout of the structure of the blood vessels. So angiogenesis is the development and growth of new blood vessels. Very, very important for normal development. As you grow and get bigger, or as you develop in utero, angiogenesis is huge. It's one of the major developmental processes that occurs during in utero and during every stage of the life cycle in humans. For instance, important in wound healing, angiogenesis can and does contribute to wound healing, can and does contribute to the endometrial growth. That's a major angiogenic. Much of the endometrial tissue is in fact blood vessels and capillaries. And it can enhance, so if you work out and you exercise regularly, what you will see is an increase in density of blood vessels in and around your heart and in and around your smooth muscle, and it can be a dramatic difference. It's actually quite stunning. 1 month or 2 months of exercise can cause a 3- or 4-fold increase in vascular flow to tissues that are regularly exercised. But like all things in life, it can be a two-edged sword. Turns out that if you took a pencil, and you can do this as an exercise, take a pencil or a pen and draw the teeniest, tiniest dot you can make on a piece of paper. In order for that dot to grow any bigger than that, if let's say that dot was a tumor, for it to grow any bigger than the smallest possible dot you could draw, that tumor has to induce angiogenesis. In fact, angiogenesis has become a major target of anti-cancer therapy, because if you can block the blood supply, you can block tumor growth. And there have been some relatively dramatic examples of anti-angiogenic therapy. Not so much in the malignant side of it. Malignant tumor cells are a tough nut to crack. I'll give you that. But in some of the more differentiated tumors, like for instance one called a hemangioma, which is an interesting one, hemangiomas are primarily an angiogenic tumor. And it turns out that you can actually block that signaling using anti-angiogenic factors. But angiogenesis is one of the major aspects of tumor growth, and it is an active area of anti-cancer research. Angiogenesis is fundamentally driven by metabolic need, or the perceived metabolic need in tissues. What I mean by perceived metabolic need, it means that if those tissues just start secreting cytokines for no reason, such as in a malignant situation, those cytokines will induce angiogenesis. And the two that I'd like you to keep in mind and just be able to recognize, one's called VEGF, or vascular endothelial growth factor. You can write that down, vascular endothelial growth factor, but we call it VEGF. And then another one called fibroblast growth factor. Remember that small vessels are primarily endothelial cells and or smooth muscle cells. So that's why FGF may be involved as well. That involves fibroblasts, and smooth muscle cells can and do respond to things like VEGF and FGF. But it's promoted by these cytokines, these specific two. There are others, okay, but these are the two poster children, I guess. But there are also inhibitory cytokines that will block angiogenesis for reasons that are not entirely clear yet. Maybe they shut down the signaling, or they counterbalance the signaling so you don't grow too much. Not really sure, but these were actually discovered as part of that anti-cancer effort. Specifically, a lab up in Harvard, a guy named Judah Folkman, who I got to meet a few years ago, pretty cool guy. In any case, they discovered first endostatin and then angiostatin. And these have become standard therapies for certain types of tumors, particularly hemangiomas. On the other side, we can see issues with angiogenesis that can actually mask symptoms. And gals, this plays right into your personal lives. Turns out collateral circulation isn't only triggered or enhanced by these mitogenic cytokines. They are also enhanced by estrogen. Estrogen is a mild angiogenic hormone that behaves in synergy with VEGF and fibroblast growth factor. And where do we see this? We see this in pre-infarct hearts. So if we have a heart over here and is suffering from coronary artery disease, and there's just enough occlusion there, not enough to cause a full-on infarction, if you will, nevertheless, this tissue is underserved metabolically. It does not have enough Q. Okay, we haven't talked about Q yet. I'll get there. Q means perfusion. Okay. We're going to talk about that when we get to lungs. So we don't have enough perfusion here to meet metabolic need. And so this tissue starts secreting cytokines, just like as if it was in pain. All right? And we will secrete cytokines, and what you'll see is that other vessels will grow into that area in order to restore blood supply. And this is enhanced by estrogen, which is why more heart attacks, and it turns out women are starting, as they get older, jump right back into the same incidence of coronary artery disease and myocardial infarction as men do. They start off slower and then they kick in and pretty much catch up to the point now that heart disease is among the leading, if not the leading killer in women. In any case, women tend to be more asymptomatic. They tend to have more catastrophic, previously asymptomatic myocardial infarctions by virtue of the fact that they have enhanced collateral development secondary to estrogen, as well as the pro-mitogenic cytokines. Here we have a rendition of that collateral circulation development. So we develop an occlusion, usually a partial occlusion. If it was a complete occlusion, the tissue would go into infarct and start dying. But it doesn't quite die. It's getting shocked by its lack of nutrients and glucose and oxygen and amino acids and so forth. Nevertheless, if there is enough signal from the cytokines that these collaterals can develop, thus bypassing, even in some cases, a complete occlusion. And here's a nice angiogram showing collaterals growing into, you can see the infarct right here, or the partial infarct. You can still see some dye contrast there. So a little bit's getting through. The collateral's growing into this area that has been underserved metabolically. And this figure is a reminder of just the fact that these are cytokines that are involved. VEGF, okay, fibroblast growth factor. You might have, some of you may have talked about TGF-beta in one of your review articles, or platelet-derived growth factor. All of these things are involved. They're cytokine-driven, paracrine-driven. Because we're pushing 25 minutes and I don't want to go too long with these things, I'm going to go ahead and stop on this slide right here. And I will pick up with a 1C installment of this chapter with the remaining slides for this chapter. So we've got about 15 more slides or 16 more slides. I'm going to go ahead and stop, and I will see you in video 1C. Thank you.